Here's a question that sounds simple, but has quietly humbled some of the smartest scientists on Earth. How did life begin? Not how did life evolve once it existed. That's a different question and a different conversation. I'm asking something more basic. How did you get from a planet with no life at all, just rocks, water, and chemistry, to the very first living cell? How do you cross that line? You'd think, with everything we know about DNA and biochemistry, we'd have a solid answer by now. We don't. And today, we're going to look honestly at why that gap is so much bigger than most people realize, and why, after 70 years of trying, scientists still don't agree on how it happened. This is the origin of life problem. Let's start by clearing up a misconception. We often talk about simple, single-celled organisms as if they're basically just a blob of chemicals that got lucky. They are not simple, not even close. Even the most stripped-down bacteria we know of require hundreds of genes working together just to survive. Genes that build proteins, that manage energy, that repair damaged DNA, that let the cell reproduce itself. And that's the minimal version. Researchers have spent years trying to figure out the smallest possible set of genes a cell could have and still be alive, and the number keeps landing somewhere in the hundreds, not the dozens. Inside that simple cell, you have a molecular machine, DNA, storing information the way a computer stores code, using a 4-letter chemical alphabet. That information gets read, copied, and translated by other molecular machines into proteins, which fold into precise 3-dimensional shapes to do very specific jobs. Get the folding wrong, and the protein doesn't work. It's less like a puddle of chemicals and more like a fully staffed factory, with a supply chain, a filing system, and a repair crew, all running at a scale smaller than you can see. So, when we ask, how did life begin, we're not asking how a puddle got a little more interesting. We're asking how raw, non-living chemistry produced a self-sustaining, self-replicating information-storing system from scratch with no previous life around to help it. That is an enormous jump, and it's worth sitting with how enormous it actually is before we talk about the attempts to explain it. Here's where things get genuinely difficult, even for scientists who are firmly committed to a purely natural explanation. Life today runs on 2 main types of molecules. DNA stores the information. Think of it as the instruction manual. Proteins do the actual work, building structures, speeding up chemical reactions, running the cell's machinery. But here's the catch. DNA can't do anything on its own. It needs proteins to read it and copy it. And proteins can't be built without instructions from DNA telling the cell how to make them. So, which came first? DNA needs proteins. Proteins need DNA. It's a chicken and egg problem sitting right at the foundation of biology. In the moony-teen 60s and 70s, scientists proposed a possible way out, a molecule called RNA, a close cousin of DNA, that can, in a lab under carefully controlled conditions, both store some information and perform some chemical functions on its own. This became known as the RNA World hypothesis. The idea that RNA came first and eventually got replaced by the more efficient DNA and protein system we see today. It's a genuinely clever idea, and it's the leading hypothesis for a reason, but it comes with serious unsolved problems. Scientists have never observed RNA molecules assembling themselves from scratch under realistic early Earth conditions. Building the individual chemical building blocks of RNA in a lab requires precise conditions, and getting them to link into long functional chains without help is extraordinarily difficult. The same conditions that help build one part of the molecule tend to destroy another part. And even if you got a working RNA molecule, you'd still need it to somehow start copying itself accurately over and over before it falls apart. Nobody has demonstrated a plausible, complete pathway from simple chemicals to a self-replicating RNA molecule in the lab. That doesn't mean it's impossible, but it means the leading scientific hypothesis for the origin of life still has a significant, unresolved gap right at its center. There's a deeper issue here that goes beyond chemistry, and it's one that even secular scientists have wrestled with publicly. DNA doesn't just store molecules, it stores information. A specific functional sequence, the way letters on a page only mean something in a particular order. Random chemical reactions are very good at producing randomness. They are not, on their own, known to produce long strings of precise, functional, meaningful sequences, the kind you'd need to build a working gene. Think about it this way. Wind blowing over a beach can pile up sand into dunes. An orderly pattern, but a simple repetitive one. It cannot arrange grains of sand into a paragraph of coherent English text. Ordinary, repeating order and specified, functional information are not the same thing. And getting from raw chemistry to the second one is a much harder problem than people often assume. This is precisely why the origin of life remains genuinely unsettled science. Not a minor technical detail waiting to be filled in, but a foundational question that some prominent researchers openly admit we don't have a working answer to. I want to be careful here, because it would be easy to overstate this. The lack of a settled answer on the origin of life does not mean scientists have given up, and it doesn't mean the field hasn't made real progress. Researchers have made genuine advances, simulating early Earth chemistry, discovering how some biological building blocks might form naturally, exploring hydrothermal vents and other environments as possible starting points. But progress on pieces of the puzzle is not the same as a completed puzzle. And what's striking is how honest many scientists in this field are about that gap. This isn't a case of religious people making up a controversy. It's leading biochemists in their own papers and conferences, acknowledging that we do not yet have a demonstrated plausible pathway from non-living chemistry to the first living cell. That honesty matters. And it leaves room for a question that's not actually anti-scientific to ask. What if the reason this problem has resisted 70 years of brilliant effort is that it's not merely a hard chemistry problem, but a signal? A sign that information, meaning, and life itself point beyond blind chemistry to a mind that intended them? You don't have to abandon science to ask that question. In fact, taking the difficulty of this problem seriously, rather than assuming it will simply resolve itself, is the more scientifically honest posture to take. So, where does that leave us? We've seen that even the simplest life is a staggeringly complex information-rich system, not a lucky puddle, but something closer to a fully operating factory. We've seen the chicken and egg problem sitting at the heart of biology, and the leading hypothesis built to solve it still missing key steps. And we've seen that specified, functional information, the kind stored in every strand of DNA, has never been observed arising from unguided chemistry alone. Maybe that gap isn't an embarrassment for science to quietly wait out. Maybe it's a genuine clue. Genesis opens with these words, "In the beginning, God created." Not chemistry alone, not chance alone. A beginning with a beginner. Thanks for watching. If this gave you something to think about, consider subscribing, and I'll see you in the next one.